Hi everyone, my name is Dr. Kavitha Rajagopal. The title of my presentation is The Biodiversity of Blowfly Chrysomya Megacephala revealed by Cytochrome B gene. Introduction. Forensic entomology uses blowfly species or larvae during crime investigation to estimate post-mortem interval. Insects as evidence. When did the victim die? Can be a crucial question in cases involving death. And the estimation of time since death, also known as post-mortem interval, PMI, is the most important task for forensic entomologists. Blowfly usually arrive to dead body within 24 hours of death or can arrive within minutes in the presence of blood or other body fluids. 3 days after death, insect evidence is often the most accurate and sometimes the only reliable method in determining the time since death. Entomologists actually estimate the time since death based on colonization of a dead body by the insects that arrive first. My estimation based on entomological evidence is strongly dependent on accurate species identification. Morphological identification can be complicated due to similarity among the species of blowfly, and in certain cases, the specimen need to be reared until adulthood. It is a time-consuming procedure. To simplify and make the species identification more practical and reliable, DNA-based identification is considered. However, occurrence of cryptic taxa can cause an error in PMI estimation. Cryptic taxa, the flies morphologically are same, but genetically they are different. This difference can affect their behavior, development rate, and other biological factors that can cause an error in PMI estimation. So, discrimination of fly species based on heuristic threshold and divergence of DNA sequence have been the common approach to identify novel taxa. The effectiveness of the mitochondrial genes in discriminating forensically important insects had been well proven. While the cytochrome oxidase 1 gene is commonly used for phylogenetic studies, cytochrome B gene also found to be powerful for identifying the geographical origins or source of the species. Objectives: to study the genetic lineages and haplotype dispersal patterns of Chrysomya megacephala using cytochrome B gene. Biological data on local flies are more beneficial in PMI estimation compared to data from other countries with different environments and fauna characteristics. Methods: 74 Chrysomya megacephala specimens were used for this study. 14 samples from Perak, 25 samples from Pahang, 14 samples from Selangor, 12 samples from Kuala Lumpur, and 9 samples from Johor. The genomic DNA was extracted from the legs of Chrysomya megacephala using the iGenomic CTB DNA extraction mini kit. 4 phylogenetic analyses were performed for this study. Number 1 is Bayesian inference BI, maximum likelihood ML, neighbor joining NJ, and maximum parsimony MP. All the nucleotide sequences were edited using ChromoMass Pro 1.5. Representative haplotype sequences of the cytochrome B gene were deposited in the NCBI gene bank under the section numbers KR336662 to KR336666. The horn fly was used as an outgroup in this study. Results: sequence analysis. 4 phylogenetic analyses reveal phylogenetic trees with the same topology but with different posterior probability or bootstrap support values. Hence, only the Bayesian inference tree is presented here. The cytochrome B tree disclosed 3 genetic lineages for Chrysomya megacephala. lineages A was limited to flies originated from Selangor and Kuala Lumpur. Lineages B and C comprised flies from all studied population. Furthermore, haplotype network analysis showed 3 recognizable haplotype lineages among Malaysian Chrysomya megacephala population as shown in figure 2. An interset of the 3 genetically different groups of Chrysomya megacephala was noticed. However, the flies from the central region, Kuala Lumpur and Selangor population, also known as Klang Valley, comprise of 3 different lineages, suggested the flies are genetically diverse in comparison with the flies from Pahang, Perak and Johor. Discussion. This study also shows an overlapping of 3 different genetically divergent groups of Chrysomya megacephala population in Malaysia. The phylogenetic analysis herein revealed that Chrysomya megacephala flies from Kuala Lumpur and Selangor are high in genetic diversity in contrast to those from Pahang, Perak and Johor. This could be in support by the fact that the transport network between Kuala Lumpur and Selangor states are well developed. It makes the migration of the flies widely distributed due to natural dispersal, gene flow, mating system, and mode of reproduction and transportation through roads. Conclusion. Better knowledge of Chrysomya genetic lineages will assist in crime scene investigation by providing information on the origin or source of the blowfly. Further work with more Calliphoridae species from different states of Malaysia should be conducted to accumulate forensic entomological evidence in Malaysia. More advanced development in molecular research will raise the value of Calliphoridae fly species in criminal investigation. References. Some of the references that I use for this study. We would like to thank Mr. John Jeffrey, Madam Rumiza Abdul Rashid, and Ms. Zulaika Zulkifli for their kind support. This study was supported by University Technology Mara Grant and University of Malaya Grant. Thank you.